the basic definitions which I want to elaborate before going to the cell cycle. You all know about the genetics. What is genetics? Genetics is basically the branch of biology which deals with the study of inheritance. And what is inheritance? It is how biological information is transmitted from one generation to the next. And these are informations are carried in DNA, and the DNA which has certain information is manifest in the kinds of proteins. Means the DNA makes protein, and the protein exhibit certain characteristics in an individuals. And I want to give you an example of eye color or hair color that you observe in your daily life, that you have different eye colors. Some have dark iris color, some has brown color, some has light brown color, and others have different eye colors. There are 9 shades of different eye colors, and there are different hair colors also. It varies from brown to different shades of blacks. And other is the hair type. Means different have straight color, or different have curly hair, or different hair shapes. So the DNA has certain informations, and these informations are firstly from different kinds of proteins, and the proteins exhibit these characteristics in different ways. Means if the DNA has the characteristics, has the information which makes a protein which transfer brown eye color in individual, all the individual have brown eye color. And if the DNA has different information, means these information which make different kinds of proteins. If the proteins will make black color of iris, the eye color of yours is black. And same is the case with the hair color. It may be white, it may be brown, it may be black. And these are made possible with the help of enzymes, and that they regulate the rates of chemical reactions in organisms. And the other things, that within certain environment limits, animals are what they are by the proteins that they synthesize. It means that the individual must have characteristics which are present in the forms of proteins. So the individuals are bound to show that characteristics which the DNA make different kinds of proteins in them, and the proteins then are responsible to make certain characteristics within the organisms. And at the level of organisms, productions involves passing DNA between individuals. Means the organisms are responsible to pass their DNA into the next individual, and this possible for existing in our species, that all individuals, all organisms have a characteristic that they pass their genes into the next individual to show its existence in this universe. You will see a figure. It consists of eukaryotic chromosomes. That these chromosomes, which are only seen during cell divisions. You can't see the chromosome when a cell is not dividing. Means you can only see the chromosome when the cell is dividing. And if we magnify a small portion of chromosome, you will see a supercoil within a chromosome. And if a small portion of supercoil is further magnified, you will see a folding within supercoil. And when only a one loop of this supercoil is magnified, you will see a structure in which DNA is wrapped between different kinds of histone proteins, and there are 5 different types of histone proteins, and one of them is the linker protein, which bind the DNA into the core particle. So this makes the DNA is more complex and more compact. And further, if we magnify, the DNA is a simple double helix structure in which there are number of different kinds of nucleotides. So nucleotides make the DNA molecule. There are 4 different types of nucleotides, and these 4 different types of nucleotides are due to different nitrogenous bases they have: adenine nucleotide, guanine nucleotide, cytosine nucleotide, and thymidine nucleotide. And nucleotide is made up of, in case of DNA, it's deoxyribose sugar, 1 phosphate, and 1 nitrogenous base. Eukaryotic chromosome, I have already elaborated in the previous slide that DNA is the genetic material, exists with protein in the form of chromosome in eukaryotic cells, and these proteins are basically the histone proteins. And during most of the life of cell, chromosomes are in a highly dispersed state called chromatin. What highly dispersed state? I have already elaborated in previous slide that you can't see chromosome when the cell is not dividing. Means you can only see a complex structure which is called a chromatin. And during these times, unit of inheritance is called genes may actively participate in the formation of proteins, because genes have all the information to make protein, and protein further exhibit different characteristics. And when a cell is dividing, chromosome exists in a highly folded and condensed state that allows them to be distributed between new cells being produced. Chromatin consists of DNA and histone proteins, and this association of DNA and protein helps with the complex jobs of packing DNA into chromosomes and regulating DNA activity. There are 5 different histone proteins. Some of these proteins form a core particle. DNA wraps in a coil around the protein, and this combination is called a nucleosome. The fifth histone protein, I have already told that it is called the linker protein. It is not needed to form the nucleosome but may help anchor the DNA to the core and promote the binding of chain of nucleosome into a cylinder. Further folding and the addition of protective proteins result in the formation of chromosomes during mitosis and meiosis. And there are 2 different types of chromatin material in chromosomes. One is the inactive portion of chromosomes produce dark banding patterns with certain staining procedures and thus are called heterochromatin regions. These regions are never exhibit its characteristics in any case, and this consists about 95% of the total chromosomes, and the other 5% of the regions are more active portions of chromosome, means all the characteristics which we exhibit are the results of this region, which is the active portion of chromosomes, and it is called the euchromatic regions, and this consists of only 5%. And one thing I want to discuss further, that when we have seen the structure of chromosomes, and you will see that there is a lot of folding of DNA in this chromosome structure. There are 2 types of chromosomes. One is called the sex chromosome and other is called the autosomes. Means autosomes have those chromosomes which make the body parts of living organisms, and sex chromosomes are involved in determination of sex of the individual. Means if we want to differentiate between male and female, the sex chromosomes are important in determination of male and female, and there are different types of sex determination protocols or ways of determination of sex between the different individuals of the different species or different animals. I want to discuss 2 or 3 types here. One of the evidence of chromosome based of sex determination came from work with the insect Proteiner. It has one darkly staining chromosome of Proteiner, which is called the X chromosome, is represented differently in males and females. Means all of somatic, means body cells of males have one X chromosomes, and all somatic cells of females have 2 X chromosomes. Means the males have one less X chromosome as compared to females. So when a male forms its uh, gametes, half of the males produce one X chromosome and the other have no X chromosome, while all the females have two X chromosomes, and when it produces in gametes, one have one X chromosome, the other one is have another X chromosome. This pattern suggests that fertilization involving an X-bearing sperm will result in female offspring, and that fertilization involving a sperm with no X chromosome will result in a male offspring. This sex determination system explains the approximately 50-50 ratio of females to males in this insect species. Means half of the individuals are female and half are male. Chromosomes that are represented different in females than in males and function in sex determinations are sex chromosome, as I have already elaborated this definition, and chromosomes that are alike and not involving in determining sex are autosomes. The system of sex determination described for proteiners is called the XO system. It is the simplest system for determining sex because it involves only one kind of chromosome. Many other animals, for example humans and fruit flies, have an XY system of sex determination. In the XY system, males and females have an equal number of chromosomes, but the male is usually XY and the female is XX. So it is the male who determines the sex of the offspring. Means if X chromosome of male combined with the X chromosome of female, it is the female, and if Y chromosome of male combines with the X chromosome of female, it is the male. In birds, the sex chromosomes are designated Z and W, and the female is ZW. Means it is the female who determines the sex of the offspring, because females have two type of sex chromosome. One is the Z type and other is the W type, and all the males have only one kind of sex chromosome, it is the W type. And this mode of sex determination also results in approximately equal numbers of male and female offspring. The example which I have given before it's the insect proteinor. The female, as I have already elated produce, have two sex chromosomes, and the male have only one sex chromosome. So if one X of female combined with the X of male, the offspring is female, and if one X of female combined with no X sperm of male, it is the male. And now I want to discuss about the number of chromosomes. As or you all know that there are fixed number of chromosomes in different species. The number of chromosomes is constant within a species, and chromosome number varies greatly among species. Humans have 46 chromosomes, chimpanzee have 48, and there are different number of chromosomes in different individuals, means in different species. Chromosomes are present in sets, with the number in a set being characteristics of each kind of animal and expressed as n. N identifies the number of different kinds of chromosomes, and most animals have two sets or 2n chromosomes, means we have 46 number of chromosomes. So this is called 2n chromosomes or diploid chromosomes or diploid condition. But the gametes of ours, in sperm or egg, have half the number of chromosomes. It is called one set or n chromosomes, and it is also called haploid. Haploid means single. Some of the individuals animals have half chromosomes, like male honeybee and some rotifers, and these have half number of chromosomes. Very few animals, for example brine shrimp, snout beetles, some flatworms, and some soap bugs, have more than the diploid number of chromosomes, and its condition is called polyploidy, means 2n means 46, 3n means 46 plus 23 means, and it further increases into 4m, 5n, 6n, 7n or 8n. Now the most important question, why cell divide? As you know, cell is the site of metabolic reaction, and continuous metabolism in the cell increases its cytosolic volume, number of endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, and microbodies. And as a result of this, this nucleocytoplasmic ratio disturbed, and to restore its proportional ratio, a cell has to divide. And one thing importantly, you have to note that there are some permanently or terminally differentiated cells, such as neurons and bone cells, which do not divide. and the duration of various stages of cell cycle, it varies in different cells. Normally, 1 ton of cell cycle is usually completed in 24 hours, and it is observed through the culture of embryonic cells. And cell has cell has different phases, and it is G1 as G2 phases. In G1 phase might last about 11 hours, S phase about 8 hours, G2 about 4 hours, and M about 1 hour. And basically the cell cycle is divided into two phases. One is the called interphase, and other is mitotic or M phase. So most of the time the cell of the cell cycle, the cell remains in interphase. In cell will spend more the time in interphase, means the 23 hours it spent in interphase and only 1 hour in M phase. And as I already told you, that this time period is never constant, because different type cells of different species take different time period for completion of 1 ton of cell cycle. Means it varies from day to weeks, or weeks to months, or even years. Now the most important topic is the cell cycle. Cell division occurs in all animals during growth and repair process, and this is only the cell cycle which is able to form a complete individual from sperm and egg union. Means from zygote the complete individual is formed, and it is also because of cell division. Cell forms from one cell it forms 2 to 4, 4 to 8, 8 to 16, and goes on. And cell divide in two basic stages. One is the division of nucleus, it is called karyokinesis, and other is the division of cytoplasm, it is called cytokinesis. And before a cell divide, the cell must grow and carry out its various metabolic processes. So what is the definition of cell cycle? A cell cycle is that period from a time a cell is produced until it completes mitosis. And I already told that cell cycle is basically consists of two phases. One is called the interphase, and other called is the M phase. And interphase is further divided into G1, S, and G2 phases. And now I will elaborate what is G1, S, and G2 phase. G1 phase, it is the post-mitotic phase and take place at the end of cell division. Means the newly formed cells accumulate energy and prepare themselves for the synthesis of DNA. And during this, active synthesis of RNA and protein take place. Means the newly formed cell takes all the energy and prepare himself for further synthesis of DNA and proteins, which are helpful in making chromosomes duplication. In S phase, it is synthesis phase. During this phase, duplication of DNA and centriole take place. The duplication of DNA results in the duplication of chromosome. As you all know that without the duplication, the chromosome cannot be divided, because if we have 46 chromosome and if it divides without duplication, so we have half the number of chromosome, and if this half further divide without duplication, it is 12 or 13. So the number of chromosomes will continue to reduce, and the time will come when there is no chromosomes available for further division. So before division, the chromosome must be duplicated, and S phase is the duplication of DNA and centriole. So when the cell is divided, it should have duplicated chromosome. So half of the chromosome will go into one cell and half of the chromosome will go to another cell. And next is the G2 phase. It is pre-mitotic gap phase or invisible phase. The synthesis of RNA and protein continues in this phase, and the formation of macromolecules for spindle formation take place, and the cell prepare itself to go into the mitotic phase. So this is the more important, the most important phase, because there is a formation of materials which makes spindle formation. What is spindle formation? These spindles are very important because it makes bond with the kinetochore present on the centromere of the chromosome, and after this binding the chromosome is able to divide. So without spindle formation, the duplicated chromosome cannot be divided. This figure shows the complete cell cycle, that it comprises of G1, which is the growth phase, and the S phase, which is the DNA synthesis phase, in which the chromosome makes its copy and duplicated, and G2 phase, and I know that there is a formation of spindle fibers, and after this the cell goes into the M phase, which is the cell dividing phase, and it will further, and the mitosis phase is further divided into two. One is the karyokinesis and another is cytokinesis, and the karyokinesis is further divided into four phases, which is prophase, metaphase, anaphase, and telophase. And after this phases are complete, then there is cytokinesis, which is the division of cell or the cytoplasm, in which one cell divided into two. Interphase, I have already explained, comprises of G1, S, and G2, occupies 90% of the total cell cycle. And interphase also sets the stage for cell division, because DNA replication is completed during the S phase of interphase. That means exact copy of DNA is made, and this process is called replication. A chromatid is a copy of chromosome produced by replication, each chromatid attached to its other copy or sister, or at a point of constitution called a centromere. And the centromere is a specific DNA sequence of about 220 nucleotides, and has a specific location on any given chromosome. Bound to each centromere is a disc of protein called a kinetochore. And I have also tell about the kinetochore, because the spindle fiber eventually attach to the kinetochore. As the cell cycle moves into the G2 phase, the chromosome begins condensation, and during the G2 phase, the cell also begins to assemble the structure that will later use to move the chromosome to opposite poles of the cell. For example, centrioles replicate, and there is extensive synthesis of protein that make up the microtubules. this is one chromosome. After duplication you will see a sister chromatids, which is just like the original chromosome. So the one chromosome has only one chromatid, but the duplicated chromosome has two sister chromatids. And you will also see the centromere or kinetochore position in the chromosome. So the other important topic which I want to elaborate is the control of cell cycle. There's, you will see from this picture that there are 3 points which can control the cell cycle. Means if a cell cycle wants to stop, these 3 points are necessary in which cell cycle can be stopped. One is the G1 point, other is the G2 checkpoint, and the other one is in the M phase, which is the spindle assembly checkpoint. In G1 checkpoint, it checks for the cell size, nutrients, growth factors, or DNA damage. Means if a cell not grow in full size, or it has some nutrient insufficiency, so it cannot go further and stop in G0 stage, means the stage of no division. And if it wants to divide, and if all the parameters which are necessary for cell cycle, it can further beyond to S or G2 phase. And if there is any deficiency in G2 phase, means if the cell not proper grow in size, or there is some DNA damage happens, so the cell may rest in this state too. But if it passes from G2 state, then in M phase there is another checkpoint which is called the spindle assembly checkpoint. It means the spindle, as I know, is necessary, which attached to the chromosome and is responsible for the separation of chromosomes. But if spindle do not form, the chromosome do not replace from each other. So this is also a checkpoint in cell cycle, that the cell cycle can be stopped from here. There are different types of proteins in cell cycle, which makes the cell cycle possible. These are cycling proteins, and there are some enzymes, which is kinases, different types of kinases. as there are different phases in cell cycle, so there are different types of proteins present in different phases of cell cycle. One is the G1 cyclins, G1 S cyclins, S cyclins, and M cyclins. G1 cyclins help to promote passage through start or the restriction point in late G1, and G1 or S cyclins bind these are cyclin dependent kinases. Cyclin dependent kinases means the kinases are the enzymes which only work under the influence of cyclins. Means if cyclins are proteins are available, the kinases work. If cyclins proteins are not available, kinases are not work. So the cyclin dependent kinases bind at the end of G1 and commit the cell to DNA replication. Means under the influence of cyclin and kinases, the DNA is replicated and there is two sister chromatins of chromosome. And as cyclins binds cyclin dependent kinase during S phase and are required for the initiation of DNA replication. And there's M cyclins which promote the events of mitosis, means prophase, metaphase, anaphase, or telophase. Cyclin dependent kinases I have already explained. These are the protein kinases that are enzymes that phosphorylate proteins using ATP. So ATP are necessary for these proteins to work. The regulated activity of cyclin dependent kinases is essential for the transition from G1 to S and from G2 to M, means from interphase to M phase, and the entry of non-dividing cells into the cell cycle. The transition from G1 to S requires a set of cyclins. As I've already told that there are different types of cyclins. G1 cyclins different from those required in the transition from G2 to mitosis, where mitotic cyclins activated the cyclin dependent kinases. Cyclin dependent kinases possesses two tyrosine phosphorylation sites. It has two phosphorylation sites. One causes activation of the enzymes and the other causes inactivation. It is the feedback mechanism, because basically means when there is excess cyclins available, the phosphorylation takes place and there is an activation of enzymes, and when there is no cyclin available, dephosphorylation takes place and there is inactivation of the protein kinases. And specific kinases carry out both the stimulatory and the inhibitory phosphorylation. Means when there is possibility to go further stages, there is a stimulatory process go, and when the cell cycle has to stop, it should be the inhibitory phosphorylation to take place, because with the protein kinases when not work, the cell cycle will not further proceed. The protein kinase along with cyclin. It forms maturation promoting factor, and this thing you have to be understanding. As I already told you that cycling are more important for the process of cell cycle. When the cell divide, when the cell wants to divide, there is the concentration of cyclin must be at peak. इसको मैं उर्दू में आपको थोड़ा सा एक्सप्लेन कर दूं कि जब एक सेल ने डिवाइड करना है या डिवाइड होना है तो साइक्लिन की कंसंट्रेशन को हाई होना चाहिए. जब साइक्लिन की कंसंट्रेशन बहुत ज़्यादा हाई होगी और प्रोटीन काइनेज उसके साथ मिल जाएंगे तो क्या होगा, एमपीएफ बन जाएगा. और इस एमपीएफ को हम कहते हैं एक्टिवेटेड एमपीएफ. और ये एमपीएफ डिवीजन करा देगा, सेल को डिवाइड कर देगा, तोड़ देगा, उसको एक सेल को दो में तकसीम कर देगा. लेकिन जैसे ही नया सेल बनेगा साइक्लिन की कंसंट्रेशन ड्रॉप हो जाएगी, बिल्कुल टूट जाएगी. यानी साइक्लिन तोड़ डीएक्टिवेटेड एंजाइम्स होते हैं जो उसको बिल्कुल तोड़ देते हैं. तो साइक्लिन की कंसंट्रेशन बिल्कुल ड्रॉप डाउन हो जाएगी, बहुत थोड़ी हो जाएगी. और जैसे जैसे सेल साइकिल की फर्दर स्टेजेस आती जाएंगी, यानी G1 होगी, S होगी, G2 होगी, साइक्लिन की कंसंट्रेशन आहिस्ता आहिस्ता बढ़ेगी. G2 के बाद साइक्लिन की कंसंट्रेशन बहुत बढ़ जाएगी. और जब साइक्लिन की कंसंट्रेशन बहुत बढ़ जाएगी तब ये दोबारा प्रोटीन काइनेसिस के साथ जुड़ेगी और एमपीएफ एक्टिवेट हो जाएगा, यानी मैचुरेशन प्रोमोटिंग फैक्टर एक्टिवेट हो जाएगा और दोबारा एम फेज यानी डिवाइडिंग फेज स्टार्ट हो जाएगी. और जैसे ही डिवाइडिंग फेज ख़त्म होगी साइक्लिन कंसंट्रेशन कम हो जाएगी. तो मतलब डिफरेंट सेल साइकिल के अंदर स्टेजेस के अंदर जो साइक्लिंग कंसंट्रेशन है वो डिफरेंट रहती है. और इसी साइक्लिंग की कंसंट्रेशन की वजह से, डिफरेंट होने की वजह से प्रोटीन काइनेसिस का जो काम है वो भी डिफरेंट हो जाता है. प्रोटीन काइनेसिस तब काम करेंगे जब साइक्लिन कंसंट्रेशन एक ख़ास मिक़दार से ज़्यादा होगी और वो जब बनेगी तो इसको हम एमपीएफ कहेंगे. तो साइक्लिंग एसोसिएटेड विद प्रोटीन काइनेसिस मेक एमपीएफ maturation promoting factor. The concentration of cyclin varies in different phases of cycle. I have already told you its concentration increases in G2 phase and attaches with cyclin dependent kinases 2 and makes MPF active. So cell division start. Without cyclin, the cell enter in G0 phase. Cyclin destroyed at the end of division phase. I have already elaborated you. And the decline of MPF and the end of mitosis, and there is start of G1 phase of next cycle. So active MPF stimulates what? Active MPF stimulates, it stimulates nuclear envelope breakdown, chromosome condensation, mitotic spindle formation, or targeted protein degradation. As you see that after M phase, the cyclin degradation enzymes degraded cyclin, and there is a low concentration of cyclin. But after G2 phase, there is the concentration of cyclin enhanced, and it combines with cyclin dependent kinases, protein kinases, and make MPF active. And when MPF active, the four things happen, means nuclear envelope breakdown, means karyokinesis take place, chromosome condensation, mitotic spindle formation, which separate the chromosome into two, and different targeted proteins further degraded cycling. So this is all about cell cycle and control of cell cycle.